Started. Welcome everyone. Welcome to the third webinar of the third series focusing on the use of proteomics in clinical applications. Just for your information, this webinar is recorded and you can ask your questions to the speaker through our Q&A box. Yep. So just an overview, Education and Training Committee that have basically created this webinar series is one of the initiatives of the International HUPO Scientific Organization, which focuses on disseminating knowledge from experienced scientists to the whole mass spec community. You can follow our activities on our webpage on the website, and you can also read up on the members that basically run this ETC committee. Just to give a calendar update, there are still a few more webinars coming up, and here are the dates for your record. Please note the timings. Depending on the locations and regions, the time zones are different. And we are also leaving in free access the records of the past webinars that you can freely see on YouTube. And we also aim to write summaries of those, and the first published editorial opinion is already available on the website of Journal of Proteome Research. So please feel free to access these webinars and these publications just to get an understanding of what the ETC committee does. So focusing on today's webinar, my co-chair Deepti Jaiswal Kundu and me, Shayantini Chatterjee, are delighted to welcome Professor Edward Nice, who will be presenting on pathology, proteomics, and precision medicine. So just a little bit of an intro for him. Edward is currently an adjunct professor at Monash University, where he is the head of the clinical biomarker discovery and validation in the Department of Biochem and Molecular Biology. He's also a scientific advisor to the Monash Antibody Technologies facility. He also additionally holds a visiting professorship at Sichuan University's West China Hospital and has been associated with Macquarie University as well. Professor Nice's long-term research interest has been in biomarker discovery and validation and high throughput monoclonal antibody production, as well as validation and assay developments, with a strong translational focus on colorectal cancer. He has an established reputation in the fields of HPLC, SPR, biosensor analysis, and proteomics, and has made significant contributions in protein and peptide micropurification. More recently, he has been involved in the development of layer-by-layer nanoparticles for drug delivery using proteomic technologies. And with that, without wasting more time, I will hand over the mic to Professor Nice for his presentation. I will just stop sharing my screen for Professor Nice to share his slides.
Okay. Sorry, almost there.
Yep, it's all good.
Okay, so I think we're all there. So firstly, thank you for that lovely introduction. And I'm really pleased to be here tonight to be able to talk to you about pathology, proteomics, and precision medicine. So the key topics for discussion, I'm going to introduce personalized precision medicine, move on to proteomics, around that discuss emerging technologies, followed by the role of pathology. I'm going to then look at the potential to cure colorectal cancer as an example, and lastly, finish on looking at future directions. So this slide, this gives an example of how important personalized and precision medicine is becoming. So this shows the publications taken from PubMed over the years 2000 to 2025. I've put a few markers there in terms of milestones. So in 2000, the first draft of the human genome was released. HUPO was launched in 2001. 2014 was the first draft of the human proteome. And you can see that in 2024, there were over 11,000 publications again showing how vibrant the field is becoming. Firstly, I'd like to dispel a few myths. Just want to see if I can get it. Yes, great. So firstly, proteomics is not just mass spectrometry. It actually comprises any sensitive high throughput method of protein analysis. For example, biosensors, microarrays, nuclear magnetic resonance, X-ray crystallography, and as well as the uses of bioinformatics for analysis of the complex data generated. Secondly, personalized medicine does not just equate with genomics. Genes only indicate which proteins have the potential to be expressed, and they do not inform on expression levels or modifications. It is proteins that are the true drivers of biology, and their measurement and understanding in a spatio-temporal fashion is essential for precision medicine. Sorry. Okay, is there a difference between precision and personalized medicine? There's actually a major overlap between the two terms. Personalized medicine is the older term, but there was a concern that the word personalized could be misinterpreted to imply that treatments and preventions were all only being developed uniquely for each individual, and that would miss treatments for groups of patients. However, most people still use the terms interchangeably, including myself. So what is precision or personalized medicine? Well, this represents a paradigm shift where we move from the current one treatment fits all approach to a situation where each patient receives an optimized treatment regime, which is based on a comprehensive understanding of the patient's individual system's biology with respect to both health and disease. So as shown here in the slide, you can go from a series of patients, you then look at collect the clinical samples, you get multi-omics data, collection of data. That's then analysed. We integrate that with lifestyle, environment, and clinical data, hopefully then to be able to develop drugs for individual or specific patients. Unfortunately, one size does not fit all. So as shown here, in a number of diseases, in fact, drugs are not particularly effective, and a large number of drugs do not work in most of the population. So it's important to work out which drugs work in which patients. And of course, this is going to lead us towards personalized medicine. So the concept of personalized medicine is not new. It probably dates back to ancient Chinese or Greek medicine. So in fact, in both the cases shown here, the Emperor Xi and Hippocrates were both using aspects of personalized medicine in the way they treated and looked at their patients. So let's move forward a couple of thousand years. Again, a couple of milestones. The first draft of the human genome was in 2000. In 2004, Leroy Hood introduced the concept of P4 medicine, which was predictive, preventative, personalized, and participatory. In 2012, Mike Schneider introduced what he called integrated personal omics profiling, and I'll enlarge on that in a slide in a minute. In 2014 in America, the ISB 100 Wellness Project was launched. And in 2015, Barack Obama, who was president at the time, released what he called his Precision Medicine Initiative. In China, in 2015, the Phoenix operation was opened in Beijing, and again, I'll talk about that later. And in 2016, the Cancer Moonshot was released, again, in America. In 2018, the HUPO launched a pathology pillar, and in 2020, in China, the Pi-Hub project was launched. So essentially then we've moved from a one-size-fits-all to a personalized approach where we can now give the right drug at the right dose at the right time to the right patient. So as I said, this then was perhaps the germinal publication indicating the potential of personalized medicine. So in this particular case, Mike Schneider used what he called integrative personal omics profiling, or HUPO, which combined genomic, transcriptomic, proteomic, metabolomic, and autoantibody profiles over a 14-month period to look at his own health situation. He generated over 30 terabytes of data, and analysis revealed that he was at risk from various medical pathologies, including type 2 diabetes, which he actually went on to develop during the course of the study. This was then enlarged to what was known as the Human Personalized Omics Profiling, or HBOT. This extended the HBOT concept, where 1,000 volunteers were enrolled at the annual HUPO meetings. Volunteers were consented, and blood, urine, and tool samples collected under strict SOPs, and health details, including food habits, personal health, physical activity, and stress levels, were taken. Interestingly, because the HUPO meetings rotate between the US, Europe, and Asia, this allowed both longitudinal studies and analysis of the variance of molecular markers across a large number of participants from different ethnic backgrounds. So we have then the concept of what I call an omics pipeline, where we go from genomics, where we can analyze the DNA sequence of a genome, to epigenomics to analyze the chemical modifications that attach to the DNA to regulate gene expression, transcriptomics to analyze the genes that have been expressed at a given time point, proteomics to analyze the composition, abundance, structure, and function of the full set of proteins encoded by the genes, metabolomics to analyze the products of cellular metabolic processes, and other omics technologies such as lipidomics, interactomics, glycomics, and microbiomics. So this now gives us the potential to move from genotype to phenotype. So then in a proteomics personalized medicine situation, we can take samples, a different range of biological samples. Those are processed using systems which are optimized for individual processing. We use then a number of high resolution analytical techniques such as GCMS, LCMS, NMR. That then gives us a large amount of data which we can analyze, giving us the potential to look at biomarker discovery, new drug targets, and biological hypotheses. And then data from that can be taken through preclinical studies and drug design to go to a clinical trials phase, which hopefully ultimately will give us an FDA-approved drug for use in individual patients. So what do we mean by biomarkers? Well, a biomarker can be defined as a characteristic that is measured as an indicator of normal biological processes, pathogenic processes, or responses to an exposure or intervention, including therapeutic interventions. Biomarkers have multiple uses. We have diagnostic biomarkers to confirm the presence of a disease or subtype or medical conditions. We can have biomarkers for monitoring to assess the presence, status, or extent of a disease or medical condition, or to evaluate the response to the intervention. We can have pharmacodynamic or response biomarkers to evaluate the response to a medical condition or clinical intervention. Predictive biomarkers to identify the probability of developing a clinical event. Prognostic biomarkers to identify the likelihood of a clinical event recurring or progression in patients which have been diagnosed already. Susceptibility or risk biomarkers to measure the risk of an individual to develop a disease or medical condition in patients who have not already got indications of having the disease, or safety biomarkers to predict toxic adverse events induced by the drug medical intervention itself. There are a number of strategies we can use for biomarker discovery, which are shown here on this slide. In particularly relevant to what we're talking about today would be MS-based profiling, peptidomics, MS imaging of tissues, and protein microarrays. They can be found in a wide range of biological samples. Examples would include whole blood, serum, plasma, urine, feces, saliva, sweat, biopsy samples, cells, exosomes, cerebrospinal fluid, pleural lavage, nipple aspirates, and of course now looking at the contents of the microbiome. And very importantly, non-invasive or minimally invasive sampling is clearly preferable. We now recognize, in fact, that panels of biomarkers will generally be required, and this is due to disease heterogeneity, particularly in cancer. Because of this, as you can see, large numbers of samples will need to be analyzed. We have an increasing numbers as we go from the discovery situation, where we look at tens of samples, likewise qualification, where we look at tens of samples, verification, where we go to thousands of hundreds of samples, validation, where we now start looking at many thousands, and clinical evaluation, and particularly population-based trials, where the numbers escalate into the millions. To do that, we need what might be called protein factories. Phoenix in Beijing is an example of a big database, discovery-driven open platform for proteomics. It has 25 mass spectrometers and the Tianhai 2 supercomputer, which originally was about, I think, number 2 in the world, but now ranks about number 15. The cost of the development was over $200 million US. There are 27,000 square meters for laboratories and offices. They have the potential to actually analyze 40 proteomes a day. They have computers which have 200 teraflops of 4 PB of storage, and importantly, to support that, a biobank which contains more than 1 million samples. Importantly, the personalized medicine market is lucrative. This shows it's forecast to reach over $1 trillion US by the end of 2032. And the market will comprise hospitals and clinics, diagnostic centers, research and academic institutes, and other end users. So again, it's a really important market. But you have to remember, and it's very important, that translation and patent protection are absolutely essential. So why do we need proteomics? Well, genes can identify possible genetic disorders, such as cystic fibrosis or Huntington's disease. They can identify patients who can benefit from targeted therapies, and a classic example of this is HER2, which is associated with worse prognosis in breast cancer patients, but also predicts better responders to trastuzumab, a monoclonal antibody, which is given to treat breast cancer. But most importantly, I said it's proteins which are the drivers of biological function. The majority of the drugs we are using today are protein-based, especially monoclonal antibodies. Most current biomarkers are proteins. Importantly, post-translational modifications can regulate function. So proteomics is not just mass spectrometry, as I said at the beginning, but includes other technologies such as NMR and microarrays. And importantly, again, that the genome is static, whereas the proteome is highly dynamic. So similar to the data I showed you earlier on, this shows the rise of proteomics based on publications in PubMed. And again, you can clearly see that, again, it's mirroring advances that we are having in omics techniques and personalized medicine. So some key developments in proteomics, again, with reference points of the first draft of the human genome in 2000 and the human reference genome released in 2013. In 1994, the term proteomics was coined by Mark Wilkins when he was a PhD student at Macquarie University. HUPO, the Human Protein Organization, was formed in 2001. In 2002, Tanaka and Fenn won the Nobel Prize in chemistry for mass spectrometry. In 2005, the first human protein atlas was released. That was very much antibody-based. And in 2010, HUPO launched the Human Proteome Project. In 2012, SRM, or single reaction monitoring, was Nature Method of the Year. And in 2014, there were low stringency drafts of the human proteome. In 2020, the HPP released a high stringency draft of the human protein. As I said earlier on, 2023, Pi-Hub was launched in China. So we now have the draft human proteome at greater than 90% completion, which in the last count was some 17,874 of the 19,773 predicted proteins. So to support this, we're now seeing a range of emerging technologies. So these are going to, in fact, improve the ability we have to generate new data. We have top-down proteomics, arm mobility mass spectrometry, massively parallel mass spec, cryo-EM combined with mass spec, CyTOF, single-cell proteomics, or high-throughput microarrays. So again, all of these are going to improve the depth at which we can analyze the human proteome. So these future directions will bring improved mass spec sensitivity, improved mass spec throughput, improved sample preparation. As I said, again, there'll be a swing towards non-MS-based technologies. We're going to see enhanced use of single-cell proteomics, coupled with other platforms like Nautilus, which again will increase the depth at which the data can be analyzed, an increase of antibody-based protocols, the availability of novel libraries, increased use of structural modeling and the new technology AlphaFold, which will support that, and very importantly, increased reliance on artificial intelligence. So as I said, HUPO is clearly a key player. It aims to credibly catalogue the human proteome parts list and discover its complexity. And that includes post-translational modifications, splice variants, interactions and functions. And it'll do that by establishing agreed, stringent, reliable standards, identify at least one protein project from each protein coding gene, confirm the expression of any remaining missing proteins, and making proteomics an integrated component of multi-omic studies to advance life sciences, biomedical sciences, and precision medicine. It does this with the use of a number of platforms. So we have a mass spec platform, a knowledge base or a bioinformatics platform, an antibody platform, and more recently, a pathology platform, which was released to bring pathology to proteomics. We generate data based on either a chromosomal initiative, which is basically a cataloging initiative, or based on data generated using biology and disease initiatives. And at the bottom, you'll see a number of the current initiatives which are working at the moment. So the pathology pillar was launched in 2018, and this recognized the important role that pathology will play in supporting the delivery of proteomics-driven biomarker discovery for clinical useful diagnostic assays and ongoing development of precision medicine. As I said on my previous slides, another important agency is the Cancer Moonshot. This was established in 2016 by President Obama to establish multidisciplinary international teams to fast-track the cure of cancer. It was championed by Vice President Biden, whose son actually died of brain cancer, and therefore is very passionate about cancer and improving treatment. It's now coordinated by NIH and CPTAC, and it's addressing multiple cancer types, including kidney, brain, breast, prostate, blood, melanoma, and pancreas. It's generated 58 publications in the last 12 months. So again, very, very productive in key areas. and currently 14 countries, as shown here, are participating. The last thing of these initiatives I'd like to mention is PIHub. So this is an ambitious global initiative which was initiated in 2020, and it was funded by the Chinese government with a view to dissecting the human body into a hierarchy of digital proteomic anatomy spaces using omics technologies. It was to identify single proteins and proteoforms, their localization and interactions at the cellular level, and then ultimately extend that to organs and tissues. Importantly, it's going to investigate dynamic changes and their impacts on health in the composition and organization of the proteome in response to age, genetic changes, lifetime adaptations, and exposure to external effects. An intelligent computational basis, which is called the PIHub Navigator, will be built, and that's going to integrate proteomic and other molecular and phenotypic data to give a new understanding of human biology, facilitate disease risk assessment, and enable early disease diagnosis and optimal therapeutic intervention to assist with the development and rollout of personalized medicine. Again, at the moment, this is extending to 20 countries, over 20 countries, over 80 research groups with over 100 top experts advising on the potential projects. Okay, so let's look again now at the publications in pathology, proteomics, and personalized medicine. And again, as we've shown before, it's mirroring again the advances that we're seeing in the omics technologies. Interestingly, and perhaps not surprisingly, many of these publications address cancer. So what is the role of pathology? Well, here the concept is to reach out to clinical and diagnostic colleagues to increase the understanding of personalized medicine, to be involved in training and education, to coordinate the identification of key unmet needs in clinical medicine, to stimulate guidelines and standards for the development of fit-for-purpose validated clinical assays, to help in the establishment of state-of-the-art biobanks with associated data, to interact with international pathology organizations, diagnostic industries, and regulatory agencies, to promote awareness of best practice and coordinate access to bioresources of clinical samples and their associated data, and importantly, assist in the development and validation of key biomarkers and biomarker panels. So where are we going next? Well, the new focus will be on understanding protein function to identify new biomarkers and drug targets. This is what's been called both by HUPO and PIHub the grand challenge. It's going to build on current resources. It's going to understand the proteome in the context of a network of networks. Again, as I said, with the help of pathology to identify unmet clinical needs. It will partner with complementary consortia to expand the reach of the HPP and increase its relevance. And again, that'll be interacting with groups like PIHub. It will develop forward-thinking strategies to provide functional information for each protein. And it will be a focal point for translational researchers for mechanisms, diagnostics, and therapeutics to improve human health. So pilot studies have already begun. HUPO has launched what it calls the CP50 project to bring function to the proteome. So this relies on PPI-based function prediction and multi-omics knowledge-based functional prediction. It'll look at phenotypic screens such as CRISPR and RNA, develop biochemical and immunological assays, and again, basically, hopefully find the function of at least 50 important proteins. One way of doing this will be what's becoming known as proteomics. So in this case, you take chemicals or antibodies and you add those into a sample to look at how it perturbs particular interactions. So a large range of chemical probes are available from Target 2035, I said, again, very, very many antibodies which are well-defined and are available. We can do the comparative mapping between plus or minus the perturber, and then we can define interactors and perturbers to understand impact and function. So in the HPP grant project, again, we've looked at networks through perturbation using two model systems, the HEC293T human embryonic kidney cell line and the HCT116 human colorectal cancer cell line, again, to see what type of networks are perturbed as shown on the little figures in this slide. Similar thing then with Pi-Hub. Again, you can see that they're taking the situation of the proteins with known functions, they're using perturbation, then look at getting that data to get inference of protein function and prediction of possible biological effects and importance. So what's the elephant in the room? Well, clearly it's big data. We have problems with data size, data security, global accessibility, data sharing, data formats, and data storage. And it's actually been forecast that countries with large populations and growing economies, and examples of that would be India or China, will actually generate zettabyte to yottabyte levels of health-related data each year. And just to put that into perspective, 1 yottabyte is approximately 1,000,000 trillion megabytes. So we really are talking big data situations. So obviously that's going to need the role of artificial intelligence. Large amounts of data are now being generated, blood test imaging, genetic screening, and proteomics. And I said that data is often health data and use that to predict potential health risks and treatment regimes. And then this predictive capability will enable healthcare providers to offer proactive preventative care. This will ultimately lead to better patient outcomes and significantly reduced healthcare costs. In fact, AI is already being used, and current applications include patient diagnosis, end-to-end drug discovery and development, improved physician-patient communication, transcribing medical documents such as prescriptions, and remotely treating patients, which is distance medicine and is important in countries like Australia. So what are the potential hurdles? Well, we have to persuade clinicians to actually uptake these systems. It has the potential of generating a two-tier medical system where you have the haves and the have-nots, where poorer countries might not be able to afford both the instrumentation or the reagents used. Potential problems with patient protection and privacy, a large number of regulatory and legal issues, a problem, particularly in the current global situations, on how to actually achieve funding for multinational collaboration. We need to then get the actual data available, so we need large amounts of data. We have to rule out bias and look at data quality, and we need quality control for artifacts in the data itself. So it's still a long way to go in getting effective or full effective use of AI in the potential workflow. So on the positive side, again, AI is expected to reshape the field of pathology in coming years. As I said, a multidisciplinary approach will be necessary. The sustainability and clinical utility needs to be considered, but perhaps very importantly, it will not replace pathologists. It will just complement them. So the AI and the pathologists will be complementary. Okay, so this for the last few minutes, I'd like to talk a little bit about cancer and one of my passions is that of colorectal cancer. So for basic cancer facts, there is a 1 in 3 lifetime risk of being diagnosed with cancer. Unfortunately, detection generally occurs when disease is advanced, and this decreases the chance of successful treatment. Supporting that, more than 65% of patients with breast, lung, colon, and ovarian cancer already have metastases at presentation. So early detection is crucial for improved prognosis. This shows the current cancer Australian incidence. So as you can see, in both males and females, bowel cancer is the second most important cancer. And amongst the major diseases in Australia, again, cancer is by far and away the most important, as shown over here. So why is colorectal cancer an important target? Well, it's the third most common cancer worldwide. In 2020, there were more than 1.9 million new cases of CRC and more than 930,000 deaths. So it really is a major disease. But if detected early, it's essentially curable by simple surgical resection. If detected late, survival is typical. So if detected early, simple surgical resection, 95% of all patients are essentially cured. If it's detected late, the survival is typically less than 5 years. The current protocol is to test for blood in the stools and follow that by colonoscopy if the test is positive. The problems there are, in fact, that there are many false positives and the expense of doing colonoscopy as a population-based test is prohibitive. So proteomics, this large-scale study of structure and function of proteins in complex biological samples, is an emerging technology for use here, particularly the use of fecal proteomics. So proteomics and other omics technologies will assist in a personalized medicine approach. And I said, but better, cheap and accurate biomarker assays are urgently required for early detection, prognosis, and monitoring, and where there is a significant unmet clinical need. So this gives an idea of the progression of colorectal cancer as it goes from a benign to a malignant situation. So you can see then it goes essentially from having small polyps present in the colon to having frank adenocarcinoma and frank cancers. This tends to be associated with a number of changes. So the changes in APC, MLH1 and MLH2, beta-catenin, KRAF, BRAF, TGF-beta-R1, and BAT26. So these become, again, essential biomarkers or potential biomarkers. And very importantly, there's actually about a 10 to 15-year time frame between the initiation of frank cancer, which does give time to actually do tests and do longitudinal studies, which hopefully will lead to better prognosis. So the concept in the stool proteomics, so the GI tract runs from the mouth to the anus. So it goes in through the mouth, the salivary glands, the head, esophagus, stomach, pancreas, down through the colon, small, large intestine, and the rectum. So in terms of collection, the stool sample collection is non-invasive, can easily be done at home, and is not sample limited. As the stool is formed, it passes through the GI tract and it samples the surrounding environment. And this then gives us the large stool samples give us enough material for proteomics analysis of metabolites, potential biomarkers, and the microbiota. So the hypothesis is that stool samples will contain signature proteins and peptides relating to GI tract pathology, such as hemoglobin peptides due to bleeding or carcinogenic antigen. And these peptides may be relatively more abundant in stools than in blood. Stool samples will be easier to assay than blood, where there is a wide variation in protein abundance, and the protein is dominated by a number of abundant housekeeping proteins, such as albumin, globulins, and hemoglobin. And they might be present in the samples due to leakage, secretion, or exfoliation. So there are a number of common bowel pathologies. I said that The largest of these by far is colorectal cancer, which involves 58 and 100,000 people in Australia. But there's irritable bowel disease, which includes both Crohn's disease and ulcerative colitis, and that involves about 400 and 100,000 people, and celiac disease, which is autoimmune, which 1,400, and apologies, and C. diff infection, which is produced by the spore-forming bacterium CD, which is present in 50 over 100,000 cases. And interestingly, CD is very, very effectively cured, 95% cure rate by fecal transplantation. So the advantages of stool testing: non-invasive, doesn't require trained staff, can be performed at home, no bowel prep required, a large biomass, so it's not sample limited, and it samples the whole length of the colon. However, there are problems. There's reluctance to handle fecal material, which results in a low compliance. So education is really importantly needed, and normalization of some sort is required for the variable sample size. As I said, the poor uptake is a major problem. So this shows data for Australia. So of tests sent out, only about 50% of those are actually returned. And funnily, it's very similar levels in a large number of countries, but it is a major problem getting patients to take up the fecal testing. There have been campaigns to improve that. This is an example of a campaign which took place in Queensland. An actor called Shane Jackson, who had an incredible funny movie called Kenny, where he was basically a toilet deliverer and cleaner, and he had this concept of make number 2 your number 1 priority with a bowel screening kit. So great to see that being involved, but they're maybe not as effective as they should be. I really think you need something a little bit more dramatic, like we had with HIV, where you really see the effects of what colorectal cancer can do to a person, and most importantly, take a test before it's too late. And please heed that advice. And any of you over 50, please think about going and getting yourself a test. So take-home messages: proteomics will play an important role in advancement of personalized precision medicine. Optimized automated sample preparation will be essential for high throughput. Personalized precision medicine will impact on optimized medical treatment. This will result in improved patient care. There will be major economic benefits. AI will play a major role in the analysis of proteomics data. But very importantly, doctors will need to be trained and encouraged to use proteomics as part of their toolbox. So with that, I thank you, and I'll be happy to take any questions. Thank you.
Right. Thank you so much, Professor Nice, for that really interesting presentation. Please type your questions in the Q&A box, and we'll be able to address those
One
questions.
question. We already have one question. If you can read.
Yep. So that's from Lorenzo Torreni. He wanted to ask whether in the estimation of the data that will be generated in the next years, those derived from the study of non-human, for example, bacterial proteome, have also been considered. I'm thinking in particular of the importance of these data for immunopeptidomics and molecular mimicry field.
Okay, it's a really good question. All of the technologies I think that we're developing are going to play a role in improving the situation. In terms of many things, I think I mentioned the microbiome, and the microbiome will clearly have a role in addressing the question raised. There already is work going on looking at the immune systems and again the role of peptidomics. So again, the initial data are already being generated. I think again what we're going to see is improvements in the depth to which we can analyze that data, and also using things like AI, how we can actually analyze that data and look to get new information in biologically important pathways.
All right, thank you for that. Please put down your questions, but I can ask someone meanwhile. So, do you have any list of proteins that are um, particularly documented for colorectal cancer from reaching benign to malignancy?
Okay, that's again another great question. Look, I think, as I said, basically, I think colorectal cancer can be cured within the next, essentially cured within the next 2 years with improved technologies. If you do a meta-analysis of the data already out there, there are a large number of potential biomarkers. Now, that might be a little bit of a worry because it's you've got to work out which are the important ones and which aren't. As I said, surely we're going to be looking at biomarker panels, and those panels will contain basically, I think, what will be different respective to different hallmarks of cancer. So you might be aware of a great paper on the hallmarks of cancer, that there are a number of key hallmarks, and I think each of those is going to have the distinct biological effects. And what we should be looking for is basically subgrouping, again, using AI or bioinformatics, the proteins which fall into those different categories, and then starting to do testing to see which of those panels have potential.
Yes, that's good. Yeah, because also I think with the personalized medicine, it would be something that would very much depend on person to person, right? It would be very specific based on
You're going
each
to
gender and
get
other
two things.
factors.
Yeah, you'll get two situations. I think you'll find one, and this is a little bit what they're saying, difference between personalized and precision. You'll get one group which is going to be specific for a patient based on all of the data that you have for them. So you'll get a very individual profile. And then you might get other groups where you find these are patients who don't respond to FOLFOX or one of the conventional treatments. And you might see a group of those, sorry, a group of biomarkers which is indicative of that, which means you don't need to waste time and money giving them FOLFOX. You look for, and you then might find other markers which start to group, which they do respond to. So again, there's going to be a lot of data generated, again, by integrating the AI with the data, with the data that we have.
Okay, thank you. Just looking for any other questions. The chat box. Deepti, you can let me know if I'm missing anything or.
I think it may be a very naive question from me is because you mentioned that proteomics is just not MS. So there are new technologies already coming in the field of. I'm not actually an experimentalist. I have not much background, so I just wanted to ask you that what is your take on new technology? For example, the SomaScan and Olink. They are, this affinity proteomics branch of proteomics. So what's your take on those new technologies? How do you see the future of those new technology and how they are going to, do in future current, for example, current from 5 years onwards? So
Okay, another great question. They're clearly important technologies.
yeah.
There are several questions at the moment. One question the mass spectrometry people are asking is, did I give the correct data? I think there's probably, the answer there is a no. You don't expect necessarily to get the same data out of a mass spectrometry experiment and a data using, say, Olink or the other technology, SomaScan.
Yeah.
I think both companies have done a lot of I think, very good research, R&D. The technologies aren't, in fact, particularly new. They've been going for some time. They've been developed, I think, honestly, by people who are looking at science and not looking at making a big buck out of it. There are increasingly, I think, papers coming out which are supporting the potential of those technologies. And the companies themselves are continuing to release improvements in the technologies, both in terms of technological improvements. They tend to sell panels, or they tend to sell reagents for panels, different panels, and they're widely increasing the number of proteins which they have covered in that. So again, I think in most of these things, it's, I like to say there are horses for courses. You do some things with mass spectrometry, you do other things maybe with Olink or SomaScan or Nautilus, one of the other technology platforms. They're talking huge numbers, of course, for some of these, where you can screen very, very large numbers of samples. Cost is probably still too high to be looking at these as for population-based screening. You've really got to bring the cost of that down to a few cents. Although, as I said, I mean, I mentioned the fact that you can't use colonoscopy for population-based screening, because typically a colonoscopy costs between $500 and $1,000 per person. You're talking several $100s for some of the genetic tests at the moment. You've got to be coming down to tens of dollars for something that's going to be useful for population-based screening. And obviously, high throughput microarrays of some sort, and that could include those other technologies, will be part of that.
Okay. Yeah, I think it's still debatable, and like you said, it is going to be, you have to see in the long run how they are going to do. And yeah, thank you for the answer, because this is what I get mostly when I ask this question, and I get to learn, and it's quite debatable. Some are more biased towards it, some are more still like to more established one, which is like mass spec. So yeah, thank you for the answer. Yeah.
It's okay.
I think we can take one more question. So the question could ask is, how do you think the single cell proteomics technology would affect the personalized and precision medicine?
Okay, another good question. It's clearly okay. The good thing about single cell is, I said, it enables us to address heterogeneity. So we can set heterogeneity, specific cell types. That's really important, I said, particularly in diseases like cancer. The techniques for generating single cells are improving. We're just at the moment in the working up the latest JPR issue, and it's clear that, again, looking at the publications coming through, that single cell proteomics is one of the increasing or more prevalent uptake in technologies. So more and more technologies are relying on single cell proteomics. Again, you'll see that, as I said, linked in with other technologies. So again, looking at it in that kind of respect as a technology in its own right, and probably the slide I didn't, but it's actually one of the new BD-HPP initiatives. So there is now a single cell proteomics initiative, and also an AI initiative. So those are two, again, I think signal the importance of those actual technologies at the moment. So yeah, I think single cell proteomics is important. It's going to continue to get more important, and it's going to continue to get to increased use.
All right, thank you for that. And you're reaching the hour. So if there's no other question, let's thank Professor Nice for such a wonderful presentation. And thanks for all of you for joining the webinar. Please stay tuned for the next webinar coming up, and you'll find details on it on the website. And would like to thank my co-chair, Deepthi, and also HUPO office for helping with all the logistics for this session. Thank you all,
Thank
and yes, see you next time.
Thank you. And let me echo my thanks to everybody as well. You've been great guys to work with. So thank you very much indeed.
Thank you.
you.